Support for Knowledge Stream is provided in part by a generous gift from the Appold Family Charitable Trust.
Thank you very much, Dr. Bilisi, for the kind invitation and the introduction. I'm very excited to be here to share some of our work during the last couple of years in my laboratory. So the light is central to everything, and my lab works, most of my lab work is actually revolved around light. So we study vision, processes associated with the cardiovascular diseases, neuronal transmission, and how the proteins, lipids work in this context. And also we study, how cancer cell migrates, and so on and so forth. So this is the outline of my talk. So first part of my talk, I will tell you how do we hijack chemically control event using light. For instance, people take beta blockers to reduce heart rate. So our effort to see whether we can replace beta blockers using light. The second part of the project, I'm going to tell you about health implication of cholesterol lowering medication, also known as statins. And finally, this is our highly publicized work on blue light excited retinal, a molecule in the eye, how that induces cytotoxicity. We believe this is a potential mechanism for the blue light associated vision loss. So I wanted to give you a take-home message about this concept called fluorescence. If you look at a structure of a cell in a textbook, you will see that. So you have a nucleus and then this Golgi body. And if you use fluorescence molecule and look at a cell, it looks like this. And here, the green color shows the plasma membrane, a protein associated with the plasma membrane. Those red dots, as well as this yellow region, orange region in the cell is representative of Golgi. So we have to use, cells are transparent. You don't see, if you look at anything, all the materials is pretty much transparent, so you don't see much. So you have to label. So this is how you see a cell when you fluorescently label it. So this is a 3D image. You can see how these molecules in real time in a cell is located. This black region in this part is the nucleus, which is completely transparent, so we don't have a nuclear marker, so you don't see it. So fluorescence, it's a very simple concept. So you have this fluorescence molecule we call GFP, or green fluorescence protein. I'm going to show you it in a minute. So what it does here is it absorbs light of one color. So it, we get excited due to a variety of reasons, right? And so some molecules get excited like that too. So you shine light and it gets excited, and it goes to, electrons of that molecule goes to excited state. They can go to a variety of excited states. And most molecules after that simply release that energy, but you don't see any color because that energy release is not radiative or color emission. But fluorescence molecule is special. All these energy levels up there will come down to, electrons will come down to the first excited state, and they accumulate right there. And then suddenly they come down to the ground state. And this energy release is the light emission. So excitation, absorb light, emission, release energy. Very simple process. So why do we need that? Because excitation, you provide more energy, and when these excited state electrons from this upper excited state coming down, they release some of that energy. So you lose energy. So energy and the wavelength are inversely proportional, mean higher the energy, lower the wavelength. So when you lose the energy, that means you increase the wavelength. So here, if you excite this molecule using blue light, it will release energy in the green light. So that allows us to see things. So that's the principle of fluorescence. So this shift we call stalk shift, but basically always it going from the blue to red. So in a microscope, when we do this, so you have a light source that emits all colors of light. And so we have a mirror that reflects that light into your fluorescence molecule containing cell. So cell absorb that light, for instance blue, and emit green light, and then green light is now going out. So this filter is very special. It will reflect blue light, but it will transmit green light. So that will allow you not to see your excitation light, which is a lot of light. You don't want, if you see all the light there, you're not going to see anything. So this will allow you to only take out the light coming out of this molecule. So this is the basic principle of fluorescence. So we use this concept to see biomolecules in living cells. So the light fuels pretty much everything in Earth. Whatever we do, right? The food, the gasoline, everything is fueled by the light. And fueled by this, we undergo a variety of different emotions, right? The love, and then you get heartbroken, and then you cry, and then people commit sins. So my lab also deals with very similar issues, but with slightly different spellings. So the law we talk about here is the light oxygen voltage sensing domain. It's a protein in plants that help them to undergo this process called phototropism, mean moving towards light. So CRY stands for cryptochromes, help blooming of flowers, and opsins are the photoreceptors in our eye. From us to jellyfish, all the way from higher organisms to the lower organisms, we can see opsin photoreceptors. The unique thing about this is their chemistry. So Lau domain has this light harvesting chemical molecule we call light harvesting antenna called FMN. And CRY has the FAD chromophore. And opsin has this molecule called retinal. You may have heard about retinal. They are contained in skin lotions and stuff. So they help. These molecules are nothing if they don't have these chemical light harvesting antennas to absorb light. So that's how we are able to see. So the first part of my talk, the hijacking chemically controlled events using light, I wanted to give you a little bit of introduction how the light sensing photoreceptors work. It's very simple. So here is the structure of the eye. So you have the cornea here, lens, and then you have the photoreceptor containing retina right here. This is back of your eye. So human eye is very unique structure where it has the signal carrying neuron in the front. Those neurons are the ones that receive light first, but they are not responding to light. And then you have these bipolar cells which connect these neurons with the photoreceptor cells. Photoreceptor cells are almost at the bottom, just above the pigment epithelium. So photoreceptor cells receive light coming through all these cell layers. If you take the eye of a squid, it's different. So their photoreceptor cells are at the forefront, and humans, it's a protective mechanisms against extensive light exposure. So we have our photoreceptor cells buried much deeper inside. So photoreceptor cells are two types. One is rod. These cells help you to see under twilight, low light conditions. And cone photoreceptors help you to see under bright light conditions. So cones are responsible for color vision. And in this, you can see these striations. So this is a mechanism to increase the surface area. So this is kind of, discs. We call them disc membranes. So here you have a structure of rhodopsin, which is a twilight photoreceptor. And so it receives light, and then it activates this internally bound bunch of proteins. And that's how we sense light. So, you open your eye, you see things, but it's a very sophisticated, complicated process. So our ambition, our goal is to see if we can substitute chemical sensitive receptors using light receptors, because light sensitive receptors belongs to this family of great receptors called G protein coupled receptors. G protein coupled receptors are very unique. They control pretty much most of the function in your body. Your heart rate, insulin secretion, neuronal transmission, immune system function, they all are controlled by G protein coupled receptors. So the 40% of the drugs in the market targeted to control these folks. So therefore, we wanted to see if light sensitive GPCRs, if the light sensitive receptors are GPCRs, so we wanted to see if we can use those receptors to substitute for the chemical sensitive GPCRs like beta adrenergic receptors, which increase your heart rate. So this is how GPCR work. This is probably the deepest chemistry I would go. Bear with me. So here a membrane, so cell goes like that, and you have your receptor here. So this is the GPCR. It could be the light sensitive or your beta adrenergic receptor. So internally, they're interacting with this trimeric protein alpha and beta gamma. So when you add, say, epinephrine, you activate this receptor, this alpha and beta gamma separates, and then they activate variety of things in the cell. In my lab, some time ago, we discovered that this beta gamma subunit not only stay on the membrane, they can actually move into internal membrane. And this move, it really doesn't work. So I'm going to skip this for a second, and I'll show you how this beta gamma translocation in a cell works. Let me see. Okay, that's good. So here is the beta gamma subunit on the plasma membrane. So when I activate receptors, see what happened to that green color. Do you see that green color is moving inside the cell? So that means you activate that membrane-bound receptor, beta gamma liberated, now they're moving into the cell. So fluorescence allows us to visualize this process in a living cell. Okay, let's move back. So when we go back to the light-sensitive G protein coupled receptors, now I use light to see what's going on. What I wanted to do is light to control what is going on. So two things, right? So if you wanted to control light, so my receptor should not be activated by the imaging light, right? So you shine some light. If you image the cell using a color of light, if that light activates your receptor, you lose the purpose. So you wanted to have a specific color for activation and specific color for imaging, right? So we were looking at these light receptors that absorb blue light to red. So here is the blue light sensitive receptor. It absorbs light all the way from UV to about 500, mean green color. And above 500 nanometer, it doesn't absorb. So our strategy was we will use receptors activation, we will use blue light, and we can use these colors of light for visualizing what's going on. So separation of these colors. So in the photoreceptor, you have this molecule called retinal. Every day you produce milligrams of retinal in your retina, and it forms a very strong bond with the opsin we call a covalent bond. When it absorbs light, it goes from this cis isomeric form to the trans here. And this chemical change is how this chemical change is sensed by the receptor and then activate the G proteins inside the cell. So here is the retinal inside the cell, inside the receptor. I'm looking at the receptor from down, and you can see upon absorption of light, retinal changes structure, and the entire receptor changes structure. So the protein bound to this receptor also sense that change. So this is the light reception process. So the GPCR, as I said, the largest drug market, I call them the sniffers because they sniff the surroundings of cells. If a chemical secreted by bacteria is coming or tumor cell is coming, or insulin is coming in there, so these GPCR can sense them, and they activate these G proteins. By doing that, they control a variety of different processes, from vision to immune response to cardiovascular, pain, addiction, and disease processes including cancer. So therefore, it's very, very crucial we understand how these receptors work. So the idea is, can we use light-sensitive GPCR, we also call them opsins, to control these functions, right? So that's the ultimate goal. So first we wanted to see if light-sensitive GPCRs can be engineered to induce that translocation thing you saw. You saw that green color moving into the cell. So we wanted to see that was induced by a chemical. We want to see you can do it using light. So here, that beta gamma subunit, that moving protein has a red color tag. I'm going to shine blue light in this part of the cell. You will see this area becoming cloudier. Let's see if I can play. No, unfortunately it doesn't play. But what you would see if this were to work, so this area would become cloudier, and when I shine blue light in this area, when I shine blue light in this area, so this area become cloudier. So here it shows the process. So here is the top. When you activate the top part intensity goes up, but the bottom intensity doesn't go up. When you activate the bottom or shine blue light in the bottom intensity goes up, but the top intensity goes down. So this allows you to spatially control where you activate signaling in a small single cell. Imagine this is, I don't have, but this is about 10 micrometer the distance. So you cannot use any other method to have such a precise spatial and temporal control. When you turn the blue light off right here, the activity goes away. So you turn on signaling wherever you want, and you turn off whenever you want. So that's the unprecedented advantage of our method has. So now, with this, we show that we can control signaling, but the problem is, not the problem, the question is, can we use opsin to control cell behaviors? So you know, these are much higher organisms, right? They are able to follow a laser pointer. So we wanted to see, so this is how actually immune cells work. Immune cells sense chemicals secreted by bacteria and tumor cells, and they follow those chemical paths, like, you know, detective following evidence kind of scenario. So here, what we did was we hijacked that cell migration pathway using a light-sensitive receptor. And here I am visualizing or imaging a lipid that drives cell migration, a phosphatase that works against cell migration in a migratory cell. You will see a red box appearing here, which is the blue light. So these cells have a little bit basal motility. And now your red light, and you see cell is kind of diving down. You can see the lipid accumulation at the leading edge of the cell, and the phosphate is also following. And you turn the lights off, there is no memory, cell stop migration. So this cell is designed by the nature to follow chemical gradients. Now it's following a light. So here is two macrophages. Macrophages, you have hundreds of thousands of macrophages in your lung. These two macrophages, we introduce a light-sensitive GPCR with the M-cherry tag. I hope this works. And I'm going to shine light, so I can fool the cells to think that there's a chemical coming in that direction. You can see those two cells are following light. And if you wanted to divorce the couple, you can pull them apart. You can see one is moving one direction, the other one is moving the other direction. So using these processes, and also we were able to control neuronal development using light. So here is a neuron, early stage neuron from a mouse brain. And at the beginning, you can see they try to grow, but we wanted to see if we can reprogram the neurite outgrowth of this neuron using light. You can see there is a tiny growth coming in this area over time, like that. And I shine light here. Now that growth is moving in this direction. And then I move my light to here. Now you can see this area start growing. You can see it's becoming bigger. And then we're going to move here, this light input. And you see now this growth is going to be shifted to this area. Now you can see it's growing into that optical input. And then we're going to try something different here. We're going to move this blue light into this area. This new light did not respond. Then we move to that. And you see now it's growing. And then all these growth are retracted when cells prioritize what they wanted to do. They don't have enough energy to perform all the growth simultaneously. Now you can actually move this and optically direct the new light outgrowth. So these demonstrations, so we published in Proceedings from National Academy of Sciences a couple of years ago. So opsins or this GPCR allow you to control these major cellular events. And the problem is, can we control any protein using light? It's just opsins? So that's another question. So this is where this cry and loud domain comes in. So this cry and loud domain, this is about the cry. They are amazing molecules. So cry has two domains. One is called cry, cryptochrome interacting protein. The other one is the cry. So this one is light sensing. And when you shine light, this undergoes a change, and then it can bind to this molecule. So if I attach a protein, whatever you like, maybe a kinase, maybe a phosphatase, anything, and when you do that, you can target it into wherever the CIBN. And now, if this protein has any, activity partners on the membrane or in the nucleus or on ER or Golgi, you can activate them like that. So if you look at this, so this is cry in the cytosol before shining blue light, and after shining blue light, now it moves to the membrane. So here is the real-time image. You shine light. At the beginning, it was all in the cytosol. Now it moves to the outside. So using this method, unfortunately, these two movies doesn't work. But what we did was we attached a cell migration inhibitor to that cry. So opsin activates cell migration, but here is a cell migration inhibitor. And then what we did was we activate cell migration pathway using a ligand. We just dump the ligand into the cell, so all the receptors activate globally. If you do that, cell wouldn't move any direction because there is no gradient. But we wanted to optically target the inhibitor to one part of the cell to see whether if cell can move away from the light. So here, I'm shining light here, and now you see cell is actually crawling away from the light. These two movies are beautiful, but for some reason the computer refused to play those. All right, so these methods are very valuable. So you can activate signaling in a small subcellular region of single cell. So we published large number of papers using this strategy, all the way about the vision to the cell migration to the cancer metastasis. So the second part of my talk, away from that optical control, we are moving into this very familiar topic about the statins or cholesterol lowering medications. So cholesterol is a vital component of cells. You need cholesterol for your cell membrane function. And however, too much cholesterol can block your arteries, induce atherosclerosis, and a leading cause of cardiovascular diseases. So statins we use to prevent or reduce cholesterol. So cholesterol begins with this molecule called HMG-CoA, and statins are kind of analogs or same family of molecule like HMG-CoA. However, they have this additional component to the molecule. So instead of producing cholesterol, this kind of inhibit the cholesterol synthesis. So that's how we reduce the cholesterol. However, cholesterol has large number of side effects. So some of the side effects associated with the vision. So some of them induce blurred vision, myopic effect, and also cholesterol, statins has been shown to inhibit the short-term memory loss, abnormal liver functions, and also muscle pain, especially muscle cramps, are associated with the use of statins. Also, this is a common question, like if you take medication for diabetes, that is not a license for you to eat more sugar, right? So it's very similar, the situation, so can I keep eating donuts? So, but this is not the real reason that, statins increase the cardiovascular risk or the risk of diabetes. There are other molecular mechanisms associated with it. However, if you look at those side effects, and these side effects or those processes are controlled by GPCRs. So we wonder if cholesterol lowering medication or statins actually perturb the GPCR and G protein signaling. So here is the cholesterol, acetyl-CoA goes to cholesterol. And this pathway is pretty large, so this is a simplified view. So this is where statin acts. It prevents the HMG-CoA reductase enzyme, so you won't produce this molecule called mevalonate, and therefore you won't make any of these, and you won't produce cholesterol. Very simple. So you inhibit one crucial enzyme. So interestingly, these two lipids in these intermediate lipids in these pathways are required for membrane anchoring of G proteins, but the ones that are translocated, so they need these intermediate products. So if you inhibit that, you will reduce these intermediate products, and therefore these proteins won't be able to interact with the membrane. So this was our hypothesis. So we thought we would be able to explain some of the the deleterious effect of cholesterol lowering medications. So here is the beta gamma with that lipid anchor coming from the cholesterol pathway. If you look at the beta gamma on the membrane, you will see they are on the membrane. And if I draw a line here, and if I measure the fluorescence, you can see this is the outside, fluorescence, no fluorescence there, and at the membrane fluorescence goes up, and inside the cell not much fluorescence, right? So, and if you calculate the ratio of the fluorescence on the membrane and inside, so this is the larger value, so this is the smaller value, so you will get a ratio greater than 1. Simple, right? Because this is higher and this is lower. When you expose cells to statins, you can see your beta gamma is no longer on the membranes, all dissolve into the cytosol. So, and if you measure, there is not much difference between plasma membrane and inside the cell. So that ratio is nearly 1. So this ratio closer to 1 means you have an inhibition of G protein lipidation like that. So first we tested if this actually not just prevent their membrane anchoring but also signaling. So this GPCR activation produced beta gamma, and then beta gamma eventually can result in increase in the cytosolic or cellular calcium. So we have a calcium dye in these cells. So I'm going to activate GPCR. You will see the cells becoming brighter. So this is the calcium production in these cells when you activate the receptor. If I do the same thing for the statin-treated cells, you will see some brightness, but few cells are coming, lighting up, but nowhere near the amount of calcium produced in this cell. So that means statins are able to produce G protein-mediated calcium mobilization in the cell. So these are some of the snapshots of that. So not only, so there are two things. One is the calcium response intensity is down when you have statins, and also number of cells showing calcium when you don't have statins is about 60%, but here it's about 10%. So substantial reduction of the calcium responding cells. So both intensity as well as the number of cells reduced upon statins. And also we examine this pathway also responsible for cell migration. So here you have a control cell. We activate migration using light. You can see cell is moving pretty nicely towards the blue light. And we do same thing after treating with statins, and you can see cell is trying to move, but it cannot move very well. So we also quantified this. So the migration of the leading and the trailing edges were substantially reduced when you treat cells with cholesterol lowering drugs. And also number of cells, invasion is one of the primary processes associated with the cancer metastasis. So cancer cell move from one place to another and then invade the other organs. So we examined that after treating cells with statins, and you can see cell invasion. So here you have about 5,000 cells invaded. So this is about like less than 1,000 or 700 cells invaded right here. So cell invasion is immensely reduced. So this actually suggests that statins can help metastasis, cancer spreading cancers. The other item we tested was if statins help recovery during a heart attack. When somebody has a heart attack and your heart stops and then you get the adrenaline, so heart is trying to jumpstart. As a result of this process, adrenaline comes in, activate your beta receptors. However, this excess adrenaline also induces this beta receptor internalization. They should be on the membrane to sense adrenaline, but when they internalize, they cannot sense. So now adrenaline coming in, beta receptors are internalized, and beta receptor activation is needed for heart, continuous heart beating. So the very moment you really need your heart to beat, now receptors are not there to induce that function. So that's we call ischemia reperfusion injury. So this is causing the most damage during a heart attack. There are other reasons as well. So we wanted to see what statin does to receptor internalization. So here is this beta receptor, a sensor for beta receptor. It is on the cytosol, but when you activate beta receptor using, say, epinephrine, you can see beta receptors are, first they move to the membrane, now they are accumulating inside. So this is the internalization. They go into the cell. They cannot sense the epinephrine anymore. But when you expose cells to statins, this internalization is substantially reduced. You can see. So this also shows that statins actually not only help cardiovascular diseases by simply reducing cholesterol, it actually actively play a role in preventing this cardiac reperfusion injury. So this work just came out about a few days ago in molecular pharmacology. So okay, the last part of the talk where we're going to talk about the blue light, how blue light becoming cell killer, right? So this work, as John mentioned, reached over nearly 500 million people globally, and then, and that paper became one of the top 100 papers published during 2018. So we were very proud of this work, and we have another paper right now under revision on the same subject. So the work we do here was this. We know how vision works now. So you need this retinal molecule interacting with the photoreceptor, and then after photoreceptors use them up, they release this retinal. And if you look at the structure of the eye, our eye is able to filter out or reflect more than 99% UV light. But our eye is transparent to blue light. So if you shine UV, it won't get in. If you shine blue light, it will get in. And if you look at the absorption of this molecule, light absorption of this molecule, retinal absorb UV and blue both. However, we don't get blue in, right? But we get plenty of blue light. We don't get blue UV in, but we get plenty of blue light. So therefore, we wanted to see what does free retinal do upon absorption of blue light. So again, a little bit of chemistry. So retinal absorb light, this is our hypothesis, and goes to this excited state, and it goes to this unusual secondary excited state called triplet state. And that triplet state can transfer the energy of that excited retinal to a molecule of oxygen and make it very toxic. This molecule called singlet oxygen. Singlet oxygen can attack chemical bonds of literally any molecule. And it can also produce hydrogen peroxide, peroxynitrile, variety of oxidative stress sources in a cell. So we thought if blue light gets into the eye, we have a molecule that can absorb blue light in the eye. And if this molecule produces toxic chemical species, what would that do to the cell? So here we are looking at a lipid, a very crucial lipid for cellular function, which is on the plasma membrane. So we have a sensor that will bind to this molecule, and you can see that molecule is called PIP2, which is on the plasma membrane at the beginning. So we will add retinal to this cell, and we will expose this cell to blue light. You can see the accumulation of that sensor in the cytosol. Just retinal and blue light, no photoreceptor, nothing. And we did this with the green light, red light, yellow light, and red light. They were not able to do this. It's only blue light. And UV light, of course, did that, but we avoided UV because eyes are not getting UV light. We did the same experiment with blue light without retinal. Cell has no effect on that. So this clearly shows that the blue light and retinal together can damage the cell. So using mass spectrometry, we examine what is happening to this molecule PIP2. I'm not going to go into detail. I'll show you what's happening. We believe that one of these lipid arms with the unsaturation is peroxidated. You know, your vegetable oil become rancid, right? Just foul smell. So that's the peroxidation. So it's like these are lipids, they can become peroxidated. We believe that retinal can induce peroxidation of this molecule. We found that not only this molecule, it also damaged the molecules like G proteins in the cell. So we don't have a full estimate about the damage yet. We are continuously investigating that. And when you damage these molecules, which are crucial for cell survival, you're essentially damaging the cell. So here we are measuring what oxygen species retinal and blue light produce. So we have a sensor for this molecule called DHE to detect superoxide. So this sensor becomes brighter when superoxide is around. So when you expose this cell to blue light and retinal, you see this become brighter. And also PIP2 on the membrane is now damaged. You can see no light, nothing happens. With the light, you can see superoxide production. And also we examine nitric oxide production. So we use this sensor called DAF2DA. When DAF2DA binds to nitric oxide, it fluoresces, green fluorescence. So in this cell, this field, we have a whole bunch of cells. We incubate all the cells with retinal, and then we expose only 3 cells to blue light. So we have a very fancy, sophisticated imaging system which will allow us to do this. So you can see only blue light exposed cells become brighter. So this shows that retinal and blue light produce nitric oxide. And the mitochondria is a very crucial organelle in cells which produce energy, and mitochondrial damage can result in variety of diseases, including Alzheimer's disease and heart failure. So here we are looking at the mitochondria. They show you this granular shape. You can see these spikes because they are granulated. But when you expose cells to blue light, you can see disintegration of mitochondria. You see after blue light, the fluorescence is very much flat. And also we examine if this process can kill cells. So we use this molecule called propydiamidite. You know, when we are living, we don't take anything come in, right? All the toxic things, I think except Friday. We don't take them. And then, but when cells are dead, anything can go in. So propydiamidite can get in when cells are damaged. So here we expose cells to blue light and retinal, and after 30 minutes you can see propydium that gets in, indicating the cell death. So here is a real-time movie where you can see that PIP2 on the cells, and there are 2 cells here. I'm going to expose this cell only to blue light. You can see that cell is becoming weird and just kind of popped. So it's not the light. If you do the same experiment without retinal, nothing happens. So if you do the same experiment with green light, nothing happens. So retinal and blue light combination is damaging. And we examine if sunlight has a large amount of blue light. So we expose cells to sunlight. Even 1 minute, we can see that PIP2 sensor is moving to the cytosol. 5 minutes, most of it's gone. And 30 minutes, you can see 99% of the cells are damaged. And you can expose cells to sunlight without retinal, nothing happens really. So here we determine what concentrations are required. It's about 500 nanomolar, fairly low. If you think about, our body has continuous production of retinal in the eye and other cells. And also, we have retinal in our circulation too. So I believe the mechanisms we are explaining here may have some relationship to conditions like melanoma, where people with, when you expose your skin to sunlight, people develop skin cancers. We believe the retinal and blue light could be a leading cause for that. We also tested that any other ways for us to prevent this. So of course, everybody these days talk about the antioxidant, right? You eat fruit, vegetables, you wanted to have your defense mechanism ready to go. So we went for the most profound antioxidant people known is glutathione. It's a cytosolic antioxidant. And so we incubate cell with this antioxidant and expose cells to blue light and retinal. It couldn't do anything. So you can see still that PIP2 is moving. We literally use this PIP2 movement as an indication. However, when we use alpha tocopherol, a vitamin E derivative, we can prevent that. So this shows that this reaction is happening in a place in the cell where lipids can go in, and not in a place like cytosol where you have a lot of water. So this is our conclusion. We believe retinal is accumulated in the membranes of cells where lipids can go in and then get excited and transfer the excited energy into the oxygen. Our eyes are highly oxygenated, and that oxygen is converted to reactive oxygen species and ended up damaging membrane interacting molecules, including proteins and lipid, and eventually ended up damaging cells. So, however, if somebody asks me that I have a mobile phone I look at like 5 hours every day and it's going to make me blind, and honestly I don't know the answer, because so many things we need to consider. The intensity of the light come in, and your retinal's recycling mechanism and your antioxidant strength, and all these things can matter. So nowadays we are looking into the very low light conditions, for instance, light coming from mobile devices and computer screens, and very low retinal concentrations, and prolonged light exposure to see how we, if retinal and blue light actually can severely cause vision damage. So recently, a nurse actually from Philadelphia called me. She said that she went to a new employer, and then she was doing this epoxy, curing during the dental procedures, and within 3 years, she developed a massive macular degeneration in her eye. And she said that, "Can you please use me as a subject for your research?" So there is so much literature, history, evidence suggests that actually blue light can harmful. It's just not the mobile phone. I don't think mobile phone is so much damaging unless you look at your mobile phone, have your covers over your head, and then no other light source, and you are staring at that 4 hours continuously. That could be damaging. But regular use of computers and mobile phone, I don't personally believe, but we need to do more experiments. But people use tanning booths, people use welders, and these occupational hazards. I think certainly we need to investigate more to find out about how blue light can be damaging. So with that, so this is my group. I have an amazing group of graduate and undergraduate students. Some of them are here, very hardworking bunch. And so we have a whole bunch of collaborators. And I'm originally from Sri Lanka. It's a beautiful place. You know, if you get a chance, please go and visit. And so our work is funded by NIH. I was initially given a very good startup from University of Toledo, and then we got money from DRC, Koch Memorial Endowment Fund, and we are also being supported by the National Eye Institute. So with that, I'm sorry the initial problem with the presentation, but I'm happy to answer any questions you may have.
Support for Knowledge Stream is provided in part by a generous gift from the Appold Family Charitable Trust.